Hi everyone, I'm Priyabrat and I'm super excited to welcome you all to a new exciting video series that's on single cell annotation. In the past, I had recorded few videos that's on the Surat pipeline where we analyzed the single cell data and we came up to this stage where we obtained the single cell clusters, right? So we know that okay, this is cluster 0, 1, 2, 3, 4 like that, but we don't know what they are. So that's what the next stage of the Surat pipeline or any single cell data analysis pipeline is, the single cell annotation. And it is one of the most complex step because we have to annotate it, right? So annotation is the most challenging part, and there are many concepts are available, many tools, many approaches are available. So that's what I thought, let's create a series to cover this vast topic. And this is what I have divided the topic on the content. Like we'll start with the very foundation introduction we'll cover in this video, where we will talk about what is cell type annotation and why we need to do it, and what are the different challenges that we might encounter, and what kind of assumptions that usually people make, and what we should not assume. So those are some important foundational concepts that we'll cover, and then we move on to talk about the actual annotation process. And there are, if we categorize the tools, right, there are two approaches are there in single cell annotation. One is the marker based annotation method, and the second category are the reference based annotation. And very recently with the AI evolvement, so we are now looking, we are finding some tools which are AI assisted annotation. So that's what I will be covering all these three category of methods. And coming to this marker based annotation, it's very simple. You take the marker, you compare against a marker databases. And so we will be covering this, different marker databases, and followed by the approaches of comparison. So people see, like, we can do a very simple overlap based approach, or we can use some statistical method to find out the actual cell type. So that's where some tools, important tools that we can use, like the cluster mode, which uses hypergeometric method, and it compares in a single function call. It calls against 7 different marker databases to tell us, like, the best cell types for a given clusters. So very interesting tool. And at the same time, we have some other tools that we will be covering, SC type and EG cell type, which have their different ways of annotating these cell types. That's what we will be covering. The second category is the reference based. So basically here you are taking your reference data, I mean the query data, and you are aligning or mapping against a reference single cell data. This is same as how we are taking our genome sequence reference, and we are aligning the reads, right? The same way you take the single cell data and you align, or you map to a reference data. And so that's where there are different categories of tools are available with this reference based. Some are correlation based like single R, and we have some projection based method like Azimut, and we have also some machine learning based logistic regression models are there. Some tools like cell typist are there, which will help us to do the cell typing based on this reference data. Okay. And the third thing, like AI said, like AI assisted annotation, we have some tools recently come up, like Cell Llama, LLM cell type, and Cassia is the very recent tool that come up. They will try to cover those in this series. Okay. So with that, we will I think this will be very useful, and yeah, because this topic is really very fast, and you will be so many tools are there, so many concepts are there, we cannot cover everything. But that's what my intention is to any tools you take, don't worry, I mean you should understand the concepts. And nowadays with AI, coding is very easy, but the concept should be clear. That's what my primary aim would be towards clearing the concept, like how we need to do it. So with that little bit introduction about the series, so let's jump onto the first part, where in this video we will cover the single cell annotation, the very basics. Then in the subsequent video we will cover this approach one by one. Okay, so yeah, let's go. Okay, so this is what the single cell data analysis steps, like we have followed any pipeline, Surat or ScanPy. You do, like, you take the cells, you filter it, you normalize the cells, then you scale the genes, you find the top variable genes. This number can be 2000 or 3000 depending on the data set, but we find only the variable genes, and you take that, and then you do a PCA analysis where you reduce the data, reduce the noise, remove the noise, take the signal, and then you use that PCA space to do the clustering. and then we visualize those clusters using the UMAP or any TSNE plot. And last, once we got the clusters, we compare each clusters against all the clusters and find those important genes which differentiate that cluster from the rest other cluster. So that's where we got the markers. So now the next step comes is like we need to annotate each cell. So this will be the last stage of the pipeline. Okay, so very simple. Already we have got what is cell type annotation. Like we ran the pipeline, we know the clusters, but we don't know whether they are T cell or they are B cell or what, or are they some new rare kind of population that we don't know. That's what our objective is to not only we want to annotate, but we want to also see whether any rare cell types is also discovered. That's what we are. That's our objective is. But annotation is very challenging, it's not easy, but there are different reasons. Like one of the primary reason is like there is no perfect marker genes. Means, for example, let's say if CD4 cell is there, right, you cannot say that if in a given cluster CD4 is expressed, that means it is CD4 T cell. You cannot simply say like one gene equal to one cell type even. Okay, because a marker gene is not exclusive to one cell type. Okay, because CD4 is also expressed in other cell type, monocytes, dendritic cell, like that. So that's where it's very important to use combination of marker. Okay, so not only one marker, but in a combination of marker, and sometimes positive and negative marker. That means this positive marker is up, but the negative markers also is not there. Then that helps us to identify the cell type. So that's where different tools are available. That's what I have explained, right? Those tools take care of this. They don't consider only few 3-4 markers. They compare the markers, and based on that they try to come up with the best guess, like okay, probably this is this cell type and that type. So that's why first challenge is like no perfect marker genes. Okay, so now the next challenge is like context, very important, because same gene will behave differently depending on the tissue, disease and species. Okay, so let's take an example. Let's say developmental trajectories. Okay, let's say cell is now evolving, right? So it is, let's say, developing. So at what state that the data has been, the single cell experiment has been done, that will decide which gene is expressed and which gene is not expressed. Same way, activation state, a resting cell and an activated cells, both the gene expression profile would be different. So that's where your markers will be different. So let's say you are doing an experiment with, let's say, an activated cell, but let's say you are comparing against suppose using a reference data set and you are using resting data set, so that way you will get a different results. Okay, and again, tissue type, let's say your experiment, your sample is from, let's say, blood, but you are comparing against brain, lungs, so that will have a different. Okay, and again, disease, like because disease, any disease will change your cell expression, and that's where it will be very difficult to get the find markers. Okay, so that's why context plays an important role. So whenever you are using these databases, right, we will see you have to very careful to look into like what tissue and what whether that's healthy or that's a disease one, or whether it is from human or it's from mouse, and which tissue, those which state it is there. So those kind of things, biologically, where it should be very close to your biological data sample that you're working with. So that's why your domain knowledge will play a very important role here. Okay, the third thing is like you cannot say that this sometimes what happens, like this is you will get a big cluster, and within that you will have multiple cells will be there. For example, one cluster and multiple cell types will be there. For example, this cluster you may see an expression of, let's say, CD3D cell, as well as you will see this expression of this gene. So because this can happen that you will see that it is T cell as well as NK cell. So this is kind of mixed cluster, because your gene expression profile is so much close that like algorithm is not able to differentiate them properly. That's what you will get one single cluster, let's say cluster 0, but within that cluster, actually when you see the marker, you will see that both these genes are expressed. So now when you are using this automated tool, you might face difficulty that what is the confidence that the tool will say that whether it is a T cell or it is NK cell, or it will say what. So that's what, wherever this cluster is not clear, then we might face some challenges. Okay, and sometimes the other way around, like one cell type equal multiple clusters, right? So for example, T cell can be split into naive T cell, effector T cell and memory T cell, like that. So they will be all together, but within that you will see some subtypes. So that's also an important challenges. Now this is the technical challenges. Like sometimes the data itself is very noisy. We will see some dropouts, low expression, and batch effect. Those will those will influence the annotation steps. For example, these are the points very important. For example, sparsity, right? Many genes are not detected even when expressed. So they are expressed, but because of your technically your some issues are there instrument, you could not detect it. But that doesn't mean that that gene is not expressed, and because your that marker is not there, you will incorrectly annotate. Okay, sometimes shallow depth, like individual cells have fewer reads than the bulk RNA sequence. Number of reads are less, so your confidence is very low to call that. Okay, and your batch effect, and sometimes doublets will be there. High expression. So that's where. But these are actually during the pipeline when we follow the normalization and batch, we have to doublet reduction, those kind of thing we do, right? So this will remove that, but still this influence your downstream cell type annotation. And yeah, and this is the most I would say, because don't know incomplete biological knowledge, because whatever literature you have, based on that knowledge only we will be able to guess the cell type. So if some new rare cell population is there in your sample, or some other different kind of state is there, then it would be very difficult to annotate your sample. That's where let's say novel population, that's what I was saying, the rare cell type may lack characterized markers. And sometimes you are dealing with like drosophila, but currently only markers are like the human and mouse is there. So how will you do that, right? So that's where closest match we do and try to guess that. And yeah, and also terminology, this is very important because literature uses a different name, right? So that's where I will be showing like how we can normalize this name and while doing the coding. So T cell, T helper cell, CD plus T cell, so those are different. So how will you take that and make a common nomenclature to use it? Okay, so that's what annotation is. This is a very challenge of incomplete biological knowledge. And last is computational complexity. Okay, sometimes computationally complexity means not only the compute power, but algorithm is not robust enough to differentiate. So these are some in our algorithm challenges are there. That's why this part is very complex, this stage. Yeah. So with that, next move to a very important part where the common misconception about the cell type annotation is a very information I got from this site. It's a very excellent site which documented nice topics. So I would recommend you to go to that if you want to read more for other exciting topic on NGS. Okay. So coming back here. So some misconception is like one marker gene is not enough. Okay, right? So we say that if this marker is there, then that is done. So that's not always the true, and we always assume that whatever automation method is like, these are always correct. So no, always that's what you have to validate the tools that what you are using. Then we might think that every cluster what we Surat gave, like cluster 0 1 2 3 means definitely that's a unique cluster. No, it can happen that within that cluster you may find 3 different subtypes. So don't hesitate to analyze the big clusters. Okay. Fourth assumption is like we assume that whatever published means like it will work universally. That never happens, because I will show you like every database markers you will give you different results. So that's where you have to sit down and look into the output and then come up with a proper conclusion. Okay. Then another assumption is like this find all markers reveal cell types directly. That's not correct. We will get the genes, but they may not be the marker genes. For example, just an example, during Surut clustering, right, you may find that ribosomal genes will be on the top differential expression genes, but they are meaningless for cell type definition. So not only you cannot only take top 20 genes. When you take top 20 genes, just evaluate whether you have some random genes are there or something like that, like ribosomal genes, mitochondrial genes like that. But anyways, we have filtered those in the very beginning pipeline, but still this is an example that it's not like if it a top gene means they will be marker genes. Okay, so top 20, let's go for top 30 40, maybe a marker genes is towards the below, because of the noise on the top, the important crucial genes are below, so that's why you have to play with that as well. And yeah, so that's what the top marker alone is not sufficient. You have to evaluate it with multiple markers as well. Okay. So yep, so with that, I think we have next 4 methods will go. Like as I said, we have the marker based, and within that you can go with the very simple overlap based and statistical method, and the reference based and AI assisted based. Okay, so just quick overview, like if you go to this marker based, right? So you got this UMAP, you take a clusters and you do the top differential expression genes, which are log to fold change is high and p value is very low, and then you take those genes, and now you use your domain knowledge expert driven, and then you compare with some marker databases, and then you say that, see, in the T cell these three are the marker genes, and you check in your marker list, oh, probably these three genes are there, then you either you do manually overlap, and based on that you can come some annotation. So in the next video, when we will work on this, we will be using Jaccard similarity based approach. So I will show you how you can do a very simple overlap based approach to come to a very superficial high level quick overview of what cell types you have. Okay, so the next type is what I said, like same comparison, but here we are comparing against the same, take your gene, comparing against the marker genes, but here you use statistical method to get some scores, and that scores will give you some the confidence behind, okay, we are confidently saying that this cell type is what. Okay, so that this is, I would recommend to use this method as well. The third one is the reference. This is what the mapping. So you take the UMAP, let's say, data, and you project it on the reference data. Okay, and based on that you are guessing, like also you are just alignment kind of thing, and you are annotating. So that's where the third approach would be. And fourth one is like AI based, like LLM based, which is already learned all this text, everything, and through that also we will be able to annotate. Okay, so with that we will stop here in this video, and we will next talk about this marker based annotation approach in the next video. And yeah, so that's all I think in this video. I hope like what what we are going to see will be very interesting, and yeah, support the channel, and I will see you in the next video. Thank you.